Clicker questions for Chapter 12, Mitochondria, Chloroplasts, and Peroxisomes. 1. True or false? Respiration takes place in the mitochondria. The answer is false. Respiration is actually taking place in two different forms. You have both aerobic and anaerobic. Aerobic, yes, does take place in the mitochondria, but anaerobic takes place in the cytoplasm. 2. The majority of the genes in the human mitochondrial genome code for A. tRNAs and rRNAs needed for translation, B. proteins needed for mitochondrial electron transport, C. proteins needed for export of ATP from the matrix, or D. proteins of the citric acid cycle. The answer is A. tRNAs and RNAs needed for translation. The total number of genes that are responsible for tRNAs and RNAs are going to exceed the proteins needed for mitochondrial electron transport. Very simply, we have two genes for the 16S and 12S for the RNAs. We have 22 that are responsible for the transfer RNAs needed for translation, in comparison to the 13 protein coding sequences that we see for mitochondrial electron transport. So while there is a substantial amount of proteins, or the genes for the proteins for mitochondrial electron transport, we do have more that encode for the transfer RNAs and also the ribosomal RNAs needed for translation. 3. Where are the genes for the human mitochondrial RNA polymerase? A. In the nuclear genome, or B. In the mitochondrial genome? Specifically for the mitochondrial RNA polymerase is exclusively found on the nuclear genome, not found on the mitochondrial genome. 4. The electrical component and the chemical component of the energy stored by the mitochondrial inner membrane are both established by the movement of A. electrons through the membrane, B. ATP across the membrane, C. pyruvate across the membrane, or D. protons across the membrane. The answer is D. Protons across the membrane. Due to its dual nature of being an ion, also being positively charged, this is what's going to give both the electrical and also the chemical component of the energy stored because of the concentration gradient we see on either side of the membrane. Number 5. The electrochemical gradient across the mitochondrial inner membrane captures energy that ultimately comes from A. mitochondrial electron transport, B. photosynthesis, C. glycolysis, or D. the sun. Ultimately, it comes down to letter D, the sun. Very simply, we as organisms are going to either consume plants or consume other animals that, if their plants, have converted the sun as well as carbon dioxide into the glycerol 3-phosphate, meaning the glucose, and then it is actually going to be broken down by different types of animals or even ourselves directly to derive ATP through the actual breakdown of the glucose molecule. Number 6, glucose brings carbon atoms and electrons to glycolysis and the citric acid cycle. The carbon atoms are lost in blank, whereas electrons end up blank. A, the citric acid cycle has carbon dioxide, and electrons end up in water. Or B, found lost in glycolysis, and end up as nicotinamide adenine dinucleotide, or NAD+. They're lost in the electron transport chain, end up in ATP. Or D, lost in the citric acid cycle as carbon dioxide and end up in ATP. The answer is A. Carbon atoms are lost as carbon dioxide from the citric acid cycle, as well as electrons in the form of water from oxidative phosphorylation. So from the entire breakdown of glucose through glycolysis, the citric acid cycle, and oxidative phosphorylation, we lose carbon dioxide and also water, so we lose carbon atoms as well as electrons in the form of carbon dioxide and water. 7. Why are lipids and not polysaccharides the main form of energy storage in animals? A. Lipids are more easily digested than polysaccharides. B. Lipids have substantially more energy stored as starch. C. Lipids are stored in liver cells and more easily released to the bloodstream. Or D. Animals lack the metabolic pathways to synthesize polysaccharides. The answer is B. Lipids have substantially more energy stored as starch. Lipids are actually more easily stored as well, or stackable, than what starch is. So in most forms of animals, you want to try and decrease the amount of weight of the energy stored that you have to increase efficiency. Trying to carry on only starch is actually not a very efficient way of carrying energy stored in an organism, whereas lipids generally are. Number 8, true or false? The ATP made via substrate level phosphorylation is different from the ATP made by chemiosmosis. Is it true the mechanisms of synthesis are fundamentally different, or false? ATP is the same molecule regardless of how or where it is made. The answer is false. ATP is the same molecule regardless of how or where it's made. Number 9, true or false? Electrons are used to make ATP as they move through the electron transport chain, and they never make it to the end of the chain. Is it true? Electrons are nothing more than packs of energy. When they give up the energy, they cease to exist. Or false? The electrons are used to reduce oxygen to the level of water. They have lost most of their energy, but they still exist as discrete electrons. The answer is false. Electrons are used to reduce oxygen to the level of water. They have lost much of their energy, but they still exist as discrete electrons. Number 10, true or false? Because chemiosmotic ATP synthesis relies on the flow of electrons through the electron transport chain, chemiosmosis is considered to be a reduction, oxidation, or redox reaction. Is it true? Any reaction that relies on the transfer of electrons from one molecule to the next is, by definition, a redox reaction. Or false? The electron transport chain is a series of redox reactions, but the ATP synthase is powered directly by the flow of protons across the membrane. This is false. Technically, the electron transport chain is a series of redox reactions, but ATP synthase is going to be powered directly by the flow of protons across the membrane of the mitochondria. Number 11, the electron transport chain, negatively charged electrons and positively charged protons start on the blank side of the membrane and end up on the blank side. This is called blank. Is it A, matrix for both protons and electrons, cytoplasm for both protons and electrons, as a process called chemiosmosis, or B, electrons only to the matrix, protons to the cytoplasmic side, a process called electron transport, or C, protons only to the matrix, electrons cytoplasmic, a process called oxidative phosphorylation, or both electrons and protons across the matrix, only electrons to the matrix, and only protons to the cytoplasmic side, and is this process called an electrochemical gradient? The answer is D. Both protons and electrons are going to start off on the matrix side. While going through the actual process of oxidative phosphorylation, the electrons will stay on the matrix side, whereas the protons will be shunted to the cytoplasmic side to increase the electrochemical gradient. 12. What is the main difference between the translocase of the outer membrane and the translocase and intermembrane complexes of the mitochondrion? A. The TOM complex is located in the inner mitochondrial membrane, the TIM in the outer. B. The TOM complex is made of lipids, while the TIM complex contains proteins. C. The TOM complex generates its own electrochemical gradient. Or D. The TOM complex relies on passive diffusion for transport, while the TIM complex uses an electrochemical gradient. The answer is D. The TOM complex relies on passive diffusion for transport, while the TIM complex uses an electrochemical gradient. In particular, the TIM complex, if you remember from the intermembrane space found in the mitochondria, it's going to be positively charged on the inner membrane while negatively charged in the matrix. So the positive pre-sequence of the particular protein is going to flow through the TIM complex as going to be drawn from the positive outer membrane to the negative inner membrane, drawing in that particular protein. Whereas the TOM complex is bringing in proteins from the cytosol through the TOM complex. It does not use any electrochemical gradient since there is no charge on the outer membrane of the mitochondrion. So very simply, the pre-sequence is going to affix itself to the TOM complex, again, through passive diffusion. 13. Phospholipid transfer proteins are able to move hydrophobic lipid molecules through the aqueous space of the cytoplasm by A, adding hydrophilic amino acids to lipids, B, changing the charge on lipid from positive to neutral, C, burying the lipid in a hydrophobic pocket inside the protein, or D, remove the polluted protein from the molecule. The answer is C, burying the lipid in a hydrophobic pocket inside the protein. This is a textbook definition for the phospholipid transfer proteins. As we know, most lipids are at the very least going to be amphiphilic, so they'll have a hydrophobic and hydrophilic ends to them. But most lipids that we find are going to be hydrophobic, so they will not be able to free flow through the cytoplasm. So the phospholipid is going to basically hide the lipid in the hydrophobic pocket to transfer from one portion of the cell to another. 14. A molecule with a net charge of positive 1 will be transported across the inner mitochondrial membrane by using the blank of the electrochemical gradient. A, voltage component, B, pH component, C, both components, or D, neither component. The answer is A, voltage component. Anytime we see any kind of question with the word charge in it, we have to assume it's going to assume some kind of voltage. In this case, it's the whole basis for the electrochemical gradient, more so for the electrical portion of the gradient. So in this case, again, voltage. 15. The mitochondria has blank membrane systems and blank energy transducing membranes, whereas the chloroplast has blank membrane systems and blank energy transducing systems. Here the answer is B. So very simply, you think about the membranes we find on the mitochondrion and the chloroplast respectively. You'll read as follows. The mitochondrion has a 2-membrane system and 1 energy transducing membrane, whereas the chloroplast has a 3-membrane system and 1 energy transducing system. 16. Ribulose bisphosphate carboxylase oxygenase has 2 subunits, a large and small. The whole enzyme is composed of 8 subunits each, hence is a 16-mer. The LSU is synthesized in the stroma, and the SSU is synthesized in the cytosol and imported to the stroma. In order to efficiently synthesize a functional 16-mer holoenzyme, there must be A. An effective means of signaling between the chloroplast genome and nuclear genome, or B. A significant amount of the large subunit protein imported into the chloroplast, D. Dystrophins to correctly assemble the 16 subunits into an active enzyme, or D. Export of the large subunit mRNA from the chloroplast for translation on cytoplasmic ribosomes. The answer is A, an effective means of signaling between the chloroplast genome and the nuclear genome. Very simply, the ribulose bisphosphate carboxylase oxygenase, or Ribisco, is going to be coded on the chloroplast DNA. So naturally, good communication between both genomes of the chloroplast and the nuclear genome is going to be effective in order to effectively synthesize the 16-mer holoenzyme. Number 18, for a protein synthesized in the cytosol to be imported to the thylakoid lumen, it must have A presequence, B, a signal sequence, C, a pre-sequence and a signal sequence, or D, 2 signal sequences. The answer is D, 2 signal sequences. We need 1 signal sequence for the importation of the protein, the thylakoid, into the stroma, and that's targeted again for translocation through the second signal sequence to go through the thylakoid membrane. Number 19, all plastids begin as proplastids with a mature form determined by environmental signals and cell differentiation. This is true. This is a textbook definition. Number 20, true or false? Chlorophyll is a unique molecule in the way that energy migrates through the photosystem antenna. Is it true? The energy migrates as resonance transfer from one chlorophyll molecule to the next, not as a redox reaction. Or is it false? The energy migrates as electrons moving from one chlorophyll molecule to the next, a form of very common oxidation reduction. This is true. The energy migrates as resonance transfer from one chlorophyll molecule to the next, not as a redox reaction. Number 21, cyclic electron flow around photosystem 1 generates A, nicotinamide adenine dinucleotide phosphate hydrogen, but not ATP, B, ATP and NADPH, C, a protein gradient and NADPH, or D, glycerol 3-phosphate and ATP. The answer is C, a protein gradient and NADPH. So very simply, the plastocyanin from photosystem 1 is going to recite the electron before it's transferred over to ferredoxin to NADP reductase, which will reduce NADP plus NADPH. In conjunction, it will shunt the proton ions from the stroma into the thylakoid lumen to increase the electrochemical gradient. Number 22, the substrates for the light-dependent reactions are A, photons, water, and NADP plus, B, carbon dioxide and water, C, water and photons, or D, protons and electrons. Answer is A, photons, water, and NADP plus. Again, these are the substrates for the light-dependent reactions. Number 23, the substrate for the dark reactions, or Calvin cycle, are A, water, carbon dioxide, NADPH, B, carbon dioxide, NADPH, and ATP, or C, NADPH, glycerol 3-phosphate, and photosystem 2, or D, glucose and oxygen. The answer is B, carbon dioxide, NADPH, and ATP. Number 24, consider a chloroplast in the light. If the pH of the lumen is about 5.0, what would you expect the pH of the stroma to be? About pH 3.0, about 5.0, about 7.0, or about 8.0? The answer is D, approximately pH of 8.0. You would expect the stroma to be more basic, especially under light reactions, as the chloroplast will actively be shunting in the protons within the thylakoid lumen, which would make it more acidic by comparison. So again, under the light reactions, you're expecting the reactions to continue to take place as long as there's photons, as well as NADP plus to be plentiful. Then you would have enough reactions going on to shunt the protons within the thylakoid membrane and make it more acidic, whereas the stroma would be more basic. Number 25, true or false? The primary end product of photosynthesis is glucose. Is it true? Glucose is used to make starch, which is exported from the chloroplast and stored in the cytoplasm before it is excreted as waste. or is it false? The primary end product is glycerol 3-phosphate, which is exported to the cytoplasm and used as basis for every carbon-based molecule in the plant, and ultimately humans. The answer is false. The primary end product of photosynthesis is not glucose, it's actually glycerol 3-phosphate. This is exported to the cytoplasm and used as the basis for every other carbon-based molecule in the plant and ultimately used for ourselves. Number 26, in plants, one would expect to find peroxisomes in the bark, stem, root, or leaf. The answer is D, leaves. You also find these in seeds as well, but since seeds is not one of the possible answers, leaf is our best possible answer here. Very simply, they will coordinate with chloroplasts to metabolize the side products of the Calvin cycle. Very simply, where carbon dioxide is going to be converted into different types of carbohydrates, in this case glycerol 3-phosphate, during photosynthesis. 7. Peroxisome biogenesis requires the fusion of two distinct ER-derived vesicles, designated V1 and V2 because A. Both V1 and V2 vesicles contain a portion of the importer. B. Both V1 and V2 vesicles contain a unique set of proteins imported from the cytoplasm. C. The V1 and V2 vesicles are derived from separate regions of the ER. Or D. The V1 and V2 vesicles must be trafficked within the trans-Golgi network. The answer is A. Both designated V1 and V2 vesicles contain a portion of the importer. Bound together will form a mature peroxisome. Number 28, peroxisomal proteins are of prokaryotic origin. The answer is false. Peroxisomal proteins are of eukaryotic origin. This comes right out of the textbook. Number 29, in blank, fatty acids are oxidized in both peroxisomes and mitochondria, but in blank and blank, fatty acid oxidation is restricted to peroxisomes. Is it A, bacteria, humans, and animals? B, animal cells, yeasts, and plants? C, microbes, yeast, and parasites? Or D, single-cell organisms, fungi, and free-living aerobes? The answer is B. In animal cells, fatty acids are oxidized in both peroxisomes and mitochondria, but in yeasts and plants, fatty acid oxidation is restricted to peroxisomes. Number 30. Plant and animal peroxisomes contain the enzyme catalase. Catalase catalyzes the decomposition of A, plasmalogens, B, peroxins, C, fatty acids, or D, hydrogen peroxide. The answer is D, hydrogen peroxide. Catalase very simply will decompose hydrogen peroxide either by converting it to water or by using it to oxidize other organic compounds.